Hello everyone, this is Michael Gray speaking to you from the University of Oxford, and I'd like to welcome you to this presentation on planning and delivery of ultrasound-mediated hyperthermia for clinical targeted drug delivery. I'd like to begin by talking about the Tardox Trial, which was a phase 1 trial of ultrasound-mediated hyperthermia for targeted drug delivery to solid liver tumors. This trial was enabled by two existing technologies, a drug and a focused ultrasound or FUS device. The drug was Tardox, a thermally sensitive liposome containing doxorubicin, and it was designed to release its payload when elevated approximately 3 degrees Celsius over normal body temperature. For this trial, the drug was given systemically, after which targeted mild hyperthermia was provided by a CE-marked ultrasound-guided FUS system. The system features a fixed focus source that was translated in 3 dimensions to heat the target tumor. Drug delivery was assessed primarily through tissue biopsies, which were collected before and after FUS exposure, and these samples were split between HPLC and microscopy analyses. In 6 of the 10 patients, hyperthermia was confirmed and monitored directly using a clinically approved temperature sensor inserted through the same needle as was used for the biopsy collection. With the long-term goal of making treatments more accessible, it was also of interest to us to see if the treatments could be done without thermometry, and instead use planning models to define treatment parameters for safe targeted hyperthermia. The model we developed started with patient-specific radiological data. This was fed along with device-specific data to 3-dimensional ultrasound and heating models, which ultimately were combined to give us a treatment planning recommendation for ultrasound dose and tumor coverage, and giving us heating statistics with uncertainty bounds. These models were first validated with early patient data where thermometry was already available, and then these models were used together for all subsequent treatments, both with and without thermometry. To summarize the results, firstly, there were no FUS-related adverse effects, and there was no evidence of instantaneous thermal ablation. For drug delivery, there was at least a doubling of intratumoral doxorubicin in 7 of 10 patients, as assessed from biopsies taken before and after FUS exposure. This included 3 of the 4 patients in part 2 of the study, which was conducted without thermometry. We saw a partial radiological response in the target tumor alone in 4 of 9 patients as evaluated by the Choi criteria. And this was particularly noteworthy given that only a single treatment cycle was given. So what we can say overall is that the procedures for Tardox were feasible and safe in this 10-participant study, and that our model enabled safe prescription of ultrasound-guided focused ultrasound treatment. We are now preparing to extend the Tardox methods to a new trial called Pandox for the treatment of non-resectable primary pancreatic tumors. And doing so comes with some additional challenges, namely the high interstitial pressure that impedes drug diffusion and causes vascular collapse, and the presence of extensive fibrous tissue that acts as an additional physical barrier. The primary objective of this trial is to quantify the enhancement in intratumoral doxorubicin concentration when delivered with Tardox and mild hyperthermia generated by focused ultrasound, and we're doing this in comparison to a cohort that will receive free drug alone. We have secondary objectives to assess safety and toxicity of the treatment, to look at relative distribution of drug, and look at radiological response. I'd like to point out a few key differences between the two trials. Firstly, for pancreas, there are no ribs in the propagation path, which should simplify the transmitted acoustic field and allow us to use lower incident powers. We'll be proceeding without real-time thermometry, instead using ultrasound guidance and using our treatment planning model to prescribe the ultrasound treatment itself. And we were using a water balloon, as indicated in the lower right, to help give us a clear propagation path. New for this trial include the collection of pancreatic and abdominal tissue properties that will feed into our model. The heating model now also includes effects of organ motion, and we are incorporating strategies for improved heating efficiency. To conclude, the Pandox protocol is currently in ethics review, and our goal is to be able to treat our first patient in the first quarter of 2021. And with that, I'd like to thank you very much for watching this presentation.